My name is Julieta Trejo, and I'm a third-year doctoral student in the Department of Psychology. My mentor is Dr. Yuanbo Peng. Today, I'm going to be sharing my research titled ECT Beyond Psychological Disorders, the Anti-Nociceptive Effect of High-Intensity Electrical Brain Stimulation in the Formalin Pain Model. The implementation of electroconvulsive therapy, also known as ECT, has been used for many decades, more specifically in patients with psychological disorders who have revealed resistance to other modes of treatment. But what about ECT in the treatment of pain? In the present study, we are testing to see if ECT proves to be effective in the relief of pain through the use of behavioral testing and continuous local field potential, also known as LFP, in the formalin pain inflammatory model. For the methodology of this study, first starting with the behavioral testing, we used 12 male Sprague-Dawley rats. In the control condition, 50 microliters of 3% formalin was injected into the left hind paw, then immediately transferred to a large clear box to observe specific pain-related behaviors. In one experimental condition, 3 rats were injected with 3% formalin and then given 3 ECT stimulations delivered at a pulse intensity of 50 milliamp, each stimulation given 10 to 15 seconds apart. In the next experimental condition, also containing 3 rats, the parameters remained the same, only this time it was swapped, and the set of 3 ECT stimulations were administered first, immediately followed by formalin. Moving into our local field potential portion, we used 32 adult male Sprague-Dawley rats, and electrodes were implanted in 4 targeted areas of the brain. We did the right anterior cingulate cortex, also known as ACC, the right and left central amygdala, you'll see this as LCA and RCA, and also the right ventral tegmental area, or the VTA. And these are all areas that are related to pain processing. For the formalin model induction, the LFP signal was recorded under 3% isoflurane anesthesia for 10 minutes as baseline in all 3 conditions. Then formalin was injected after the 10-minute baseline recording in the control condition of 10 rats, also known as Group A, and also in the experimental condition of formalin followed by ECT in 12 rats, also known as Group B in this study. In the third condition of ECT administration followed by formalin in 10 rats, formalin was injected after ECT, and we continued the recording for 60 minutes post-injection. This is also referred to as Group C in the study. So to simplify what I just said, Group A is our formalin-only control condition. Group B, we did formalin and then ECT, and in Group C, we swapped this and we did ECT and then formalin. So for the ECT model induction, in the 2 experimental conditions, the ECT parameters started with a pulse intensity at 5 milliamp. We then did a 10-minute recording and then followed by a set of 3 stimulations at 20 milliamps. And we recorded the LFP again for 10 minutes. And finally, we delivered the last set of 3 stimulations. And during this last set, we cranked the milliamp up to 50. So for the results of this study, I'm going to shift to our behavioral data first. And the blue represents the formalin-only group versus the orange, which is ECT and formalin. And this pain score trend reveals a significant decrease when ECT is administered in comparison to the formalin-only group. Specifically, a significant decrease between the 30-minute to 55-minute time point between groups. And this behavioral testing data was really exciting for us in that it demonstrates the analgesic effect that ECT may evoke. So moving down to our LFP results, for the sake of time, I want to shift to our 5 minutes data, specifically just focusing on the left central amygdala delta region. And apologies if this is a little bit blurry. Group A in the blue bars, Group B in the orange bars, and then Group C in the gray bars. And Group B revealed a trend of brief inhibition of formalin-induced activity in all 4 brain regions, versus Group C demonstrates a facilitatory effect, as you can see by the increase of power, especially when compared to our formalin-only group. So the ECT-induced increase or decrease of power has a short duration of a few seconds to a few minutes, and this reveals that ECT does not exert a long-lasting effect, but rather a brief effect. So due to the Group C condition not demonstrating as salient of an inhibitory LFP effect as a Group B condition, we lead to the conclusion that administering ECT after a pain-inducing event displays a trend that it may be the most effective. So in conclusion, the behavioral and LFP results demonstrate that ECT evokes an analgesic and anti-nociceptive effect. Thank you for your time.